Can you hear me at the back? Let the bloodshed begin. So, before I tell you something about what I do, there's something I'd like to know from you. So I'm going to start with a question, just one question. How many of you in this room identify as a human? Can we have a show of hands? Come on, more people. There's a lot of humans in here. Nice to know. Well, depending on how human you identify yourself to be, you may or may not like what I'm saying next. For every single human cell in your body, there is an equal, if not more, number of bacterial cells living in you. What this means is that you are, at the very most, only half human. If you're a woman, like me, you're more likely to be a third human. Now, the other half of you is this often ignored and invisible collection of microorganisms that we like to call the microbiome. Now, it's not a bad thing, this microbiome. It's good for you. Unfortunately, it's also the reason we fart. Microorganisms are not just in us, they're also around us. If you took a walk outside and looked at the soil that you would find under your feet, a tiny gram of soil has almost 1 billion microbial cells in it. I'll let that sink in, because here I have 8 grams of soil with me. And in this, the number of bacteria I have outnumbers the total number of people living on the planet Earth. So, I'll pass it around for you. This carefully curated collection of 8 billion microorganisms. Now, for a tube that small, you'd have to think that they're probably pretty tightly packed, aren't they? Well, the soil microbiome is a very complex environment. Microorganisms, they live together in these interconnected communities with lots of interactions. They can be friends and foes. They can attack and defend. They can trade and boycott. They can collaborate, and they can compete. They may not have Facebook, but they definitely are a social network. And boy, do they talk. For a very, very long time, we have thought that these miniature monsters, they are living in isolation and working under our control. Yeast brewing our micellars, bacteria producing our antibiotics, fungi working towards our dana blue. Unfortunately, even though this might be true in industrial settings, if you take a walk out in nature, in natural environments, we couldn't be far from the truth. Microorganisms, they have a thriving social life. They have their very own Game of Thrones going on in there. On a particularly lonely weekend, I also figured out that probably the bacteria living in me have a much more happening social life than me in Copenhagen. Well, it's true. So, if these bacteria are as social as we think them to be, we came to a very important question. Can a good bacteria fall into bad company? Let me give you an example. Staphylococcus is a group of bacteria that are most commonly found in the human airway, in our throats, in our trachea, but under certain circumstances, they can be the cause of very severe respiratory infections. Fusariums, which are a group of fungi, they are the most harmless soil-dwelling organisms that you will ever find. And yet, they can also be the cause of very significant root infections that might actually kill a plant. So what is happening? How are these transitions being made from a pathogen to a beneficial organism to a harmless organism? are social interactions driving these changes? I'll come back to Game of Thrones here. Remember this house? The owners of the bridge, House Frey? They were not really a bad house. They were pretty good, doing their own thing. And then they met the Lannisters. And we all know what happened then. Some pretty horrific stuff in the form of the Red Wedding. For people who haven't watched Game of Thrones here, everyone in this picture dies. So what happened? Did House Frey fall into bad company? And can we study how this happened? What interactions caused it? So coming back to social microbial interactions, how exactly do we study them? And a very important thing that we have to keep in mind when we want to study social interactions between microorganisms is that we have to investigate them at a scale that is relevant for microorganisms. We're in a room, I think we're more than 100 here. If I were to pick a random person sitting here and say we interacted, how right would I be? I'd say fairly. You've heard my talk, we might get a beer together later, make some more fart jokes, who knows? What if I selected a random dude from Jutland? Are we ever going to come in contact? Maybe, but most likely not. And the same principle applies to microorganisms. If microorganisms are playing Game of Thrones with each other, we assume that they are living together. They are co-inhabiting a space and they have to be located right next to each other to talk to each other, to interact with each other. So that's what I did. I put on my stocking pants and like a creepy, jealous old lover, I decided to impose on their private lives. I sat at a microscope and I peeped through for a very long time. I took a very, very, very tiny particle of soil and I looked. I looked at who is sitting next to whom. Who are the neighbors? Who are the friends? Who are the foes? How often are they always together in the same configuration? And more importantly, if I take this one bacteria, does it behave the same when it's alone as opposed to when the neighbor is home? I know, it's pretty creepy, but that's what I do. What did I learn from this stalking experience? I just want to clarify, I'm not stalking people, only microorganisms. What we learned is that microorganisms are, I'm sorry to say, flaky as hell. Categorizing them as a pathogen, as a beneficial, as a harmless organism is nearly not enough. Instead, the pathogenic potential of a microorganism is much better represented in who they are talking to, who their neighbor is, and what space they sort of share with each other. We call this a pathobiome. So in my research, what we're trying to do is pretty unorthodox in that we're moving away from a simplistic understanding of one pathogen causes one disease to a more complex sociological phenomena where we think interactions between multiple partners is leading to disease development. And we call this the pathobiome, guys. If you have trouble visualizing it, the pathobiome is literally like the mafia of the microbial world. They are scary because they band together. One could also argue that I'm being paid to unnecessarily complicate our understanding of this world. And while that may be true, what is also true is that we depend on microorganisms more than ever before. For food security, for food safety, for human health, for environmental safety, and so on. We are applying microorganisms with magical properties like the ability to degrade plastic, the ability to purify water, to prevent infections, to promote the growth of our crops. We are adding these microorganisms to many different environments. And that is great. But now that we know they're a social bunch, there's something more we have to start thinking about. Does their behavior stay the same when they are there by themselves in the lab, as opposed to when they are there with a neighbor in the environment? Maybe they change, maybe they don't. Well, they can change for the better, but sadly, they can also change for the worse. So if there's something I'd like to take away from today's talk, that will be this one, that we live in a microbial world and not the other way around. That microbes have a vibrant social life. Sometimes it can be more social than ours. That three, these social interactions have important consequences for us, such as in the ability to prevent diseases, but also for their ability to cause diseases. And because you've been a really nice audience, I'm going to throw in a fourth one. If you do fart in an elevator, you can very, very confidently blame it on your microbiome. Thank you for your attention.